Hi everyone, my topic is peristalsis and defecation. We all know what is peristalsis. Peristalsis is a series of wave-like muscle contraction that move food through the digestive tract. It starts in the esophagus where strong wave-like motion of the smooth muscle move balls of swallowed food to the stomach. It was first described by Baylis and Starling Physio as a type of motility. The first step of peristalsis is swallowing of food. In the mouth cavity, food is chewed into small pieces by the tooth and mixed with saliva. The tongue rolls the food into a soft mass called bolus. During swallowing, the tongue rises to push the bolus towards the pharynx. The soft palates move up to prevent the bolus from entering the nasal cavity. The epiglottis also lowered to cover opening to the trachea. This prevents the bolus from entering to trachea. This is very important. These actions are under neural controls of several areas of cerebral cortex, including the motor cortex. Pharyngeal phase starts with the stimulation of tactile receptor in the oropharynx by the food bolus. Then the bolus enters from mouth to the esophagus. The wall of esophagus consists of circular muscle and longitudinal muscle. When the bolus enters the esophagus, the circular muscle behind it contracts and longitudinal muscle relaxes. At the same time, the circular muscle in front of the bolus relaxes and longitudinal muscle contracts. This action pushes the bolus down from esophagus to stomach. This alternate contraction and relaxation of muscle produces a wave-like movement that is peristalsis. This is the walls of esophagus, circular muscles and longitudinal muscle. It helps for peristalsis. The contraction and relaxation of these muscles produces the moment of peristalsis. This figure shows how food bolus pass through esophagus to stomach by the help of contraction and relaxation of esophageal muscles. Diagrammatic show of peristalsis movement of esophagus. Then defecation. Defecation is the act of expelling feces from the digestive tract via the anus. Defecation is also called bowel movement, that is the act of eliminating solid or semi-solid waste materials from the digestive tract. In human being, waste are usually removed once or twice daily. Feces are formed in large intestine by the process of peristalsis and stored in sigmoid colon. Muscular contraction in the walls of the colon move fecal material through digestive tract to the rectum. Act of defecation involves development of mass movement, feces pushed into rectum, and defecation relax is initiated. Defecation relax is initiated by contraction of rectum and relaxation of internal and external anal sphincters. Two sphincters control the act of defecation, the internal anal sphincter and the external anal sphincter. The internal anal sphincter consists of smooth muscle cells under involuntary control, whereas the external anal sphincter consists of voluntarily controlled striated muscle cells. The colon is responsible for propelling feces towards the rectum and beginning the urge to defecate. The external anal sphincter and the puborectalis muscle relax to allow the passage of feces out of the rectum. Abdominal muscle contraction are performed to increase intra-abdominal pressure and expel feces more rapidly. Rectal afferent nerves are responsible for the sensation of rectal fullness and the urge to defecate. Sacral nerves S2 to S4 supply innervation to the muscles most involved in the act of defecation via the pudendal nerve. പെരിസ്റ്റാൾസിസ് മൂവ്മെൻറ്റ് വഴി വേസ്റ്റ് മെറ്റീരിയൽ സിഗ്മോയിഡ് കോളനിലെത്തും. സിഗ്മോയിഡ് കോളനിൽ വേസ്റ്റ് മെറ്റീരിയൽ സ്റ്റോർ ചെയ്യും. Contraction of walls of sigmoid colon pushes the waste material to the rectum. More waste material comes to the rectum in this way. Then rectum become expanded. Pressure in the rectum increases and the rectum wall contracts. At the same time, internal anal sphincter and external anal sphincter relaxed. And abdominal muscle contraction are performed to increase intra-abdominal pressure and expel feces more rapidly. This allows the passage of feces out of the rectum. Then the internal and external anal sphincter along with the puborectalis muscle allow the feces to be passed by muscles pulling the anus up over the exciting feces. See, this is the pathway of defecation and relax. First, entry of feces by mass movement to the sigmoid colon. Then contraction of wall of sigmoid colon and push the waste material to the rectum. Rectum is distended. Sensory nerve endings in the rectum are stimulated. Impulses are transmitted via afferent fibers of pelvic nerve to defecation center. Center send motor impulses to the descending colon, sigmoid and rectum via afferent nerve fibers of pelvic nerve. Impulses cause strong contraction of sigmoid colon and rectum and relaxation of internal anal sphincter, simultaneously relaxation of external sphincter. First, peristalsis movement വഴി വേസ്റ്റ് മെറ്റീരിയൽ സിഗ്മോയിഡ് കോളനിലെത്തും. സിഗ്മോയിഡ് കോളൻ്റെ വാൾ കൺട്രാക്ട് ചെയ്ത് വേസ്റ്റ് മെറ്റീരിയൽ രക്തത്തിലേക്ക് പുഷ് ചെയ്യും. രക്തം ഡിസ്റ്റെൻഡ് ചെയ്ത് സെൻസറി നെർവ് എൻഡിങ് സ്റ്റിമുലേറ്റഡ് ആവും. Then impulses are transmitted via afferent fibers of pelvic nerve to defecation center. ഈ നെർവ് വഴി ഡെഫിക്കേഷൻ സെൻറ്ററിലേക്ക് ഇമ്പൾസസ് ട്രാൻസ്മിറ്റ് ചെയ്യും. ഡെഫിക്കേഷൻ സെൻറ്ററിൽ മോട്ടോർ ഇമ്പൾസസ് സെൻഡ് ടു ദ ഡിസെൻഡിങ് കോളം and sigmoid colon and rectum via afferent nerve fiber. This impulses from afferent nerve fiber causes strong contraction of sigmoid colon and rectum and relaxation of internal anal sphincter and relaxation of external sphincter. From there, feces is pushed out through the anus. This is the pathway of defecation. First, waste material sigmoid colonലെത്തും. Then rectum. Then waste material accumulate ചെയ്യും rectum. The rectum wall expanded. At the stretch receptors are stimulated and stretch receptor activated in rectal wall. And impulses are sent to the rectum. Rectum wall contracts. Same time, internal anal sphincter and external anal sphincter relax, and waste material comes out through anus. Topic is bulk transport. Bulk transport is a type of transport which involves the transport of large amount of substance like lipid droplets in solid food particles across plasma membrane by utilizing energy. Special processes are involved in the transport of such large quantities of materials are endocytosis and exocytosis. Endocytosis is a cellular process in which substances are brought into the cell. During this process, the plasma membrane surrounded and engulfs the particle, forming a vesicle containing ingested material. Endocytosis is a form of active transport. It includes three processes. First one, phagocytosis or cell eating. Second one, pinocytosis or cell drinking. And last one, receptor mediated endocytosis. Phagocytosis is if the material taken is large, such as bacteria or a food particle. Then pinocytosis occur vesicle form around a liquid or very small particle, so it is called cell ringing. Third, receptor mediated endocytosis. During this process, molecules accumulate in the coated pit. The pit deep seals and is incorporated into the cell as a vesicle. See, this is phagocytosis. Particles are large. This enters into the cell across plasma membrane and form a vesicle. This is pinocytosis. In this process, particles are fluid-like. Enters cytoplasm across plasma membrane and form a vesicle, membrane-bound vesicle. This is receptor-mediated endocytosis. In this process, particles are bind with the receptor and enters to the coated pit. This coated pit deep to the cytoplasm and forming a coated vesicle surrounded by coat protein. Then what is exocytosis? Exocytosis is the opposite of endocytosis. During exocytosis, membrane-bound vesicle move to the surface of the plasma membrane, fuse with the membrane and then release their contents to the outside of the cell. Exocytosis is also a mechanism by which cells are able to insert membrane proteins such as ion channels and cell surface receptors, lipids and other components into the cell membrane. Vesicle containing these membrane components fully fuse with plasma membrane and become part of the outer cell membrane. This is exocytosis. In this process, contents are moved out of the cell. Vesicles are moves to the surface of plasma membrane and fuse with membrane, and only contents are released out to the cell and it become part of outer cell membrane. Thank you.